I'm Regan Jarvis. I'm the co-founder and the CEO at Anocca. I'll just jump straight into it. So some fun facts about Anocca. Most of you probably haven't heard of us. We're a Swedish biotech working with T-cell therapy. We're established back in 2014 with an idea on paper to build a platform to decipher T-cell immunity. It's basically what we do. We're grown to 115 people, primarily scientists, a growing team of engineers and software developers in the company. R&D facility about 3,000 square meters, former AZ facilities near Stockholm. And in those facilities, we've built a platform. And it does two things. It finds targets and generates TCRs. We do a lot of things with those, but like I'll speak to today, we're focused on TCR-T for oncology, where we've got a pipeline of over 40 assets, which sounds like an enormous pipeline, and it is, and I'll describe why. We'll be clinical stage later this year with some KRAS mutant targeting TCR-T assets, and we're building those assets or manufacturing those assets in our own in-house GMP manufacturing facility. And just to mention, yeah, we've got a great backing in the Nordics. We've raised about $150 million so far. So just to say exactly what the promise of targeted cell therapies is, and when I say targeted cell therapies, T-cell therapies particularly, I mean that they've been engineered to be directed towards a target in malignant cells. CAR-Ts have redefined treatment of hematological malignancies, and they're amazing, but there is a very limited target space. That's why they're great in hematological malignancies, but have so far very limited application in solid tumors. TCR-T opens up a target space for these targeted T-cell therapies by looking at genuine T-cell targets, which I'll describe. The challenge there is that the genetic diversity in tumors and the genetic diversity among patients make the target space and the receptor space quite complex. So the barrier to entry, the fundamental barrier to entry is, can you map those targets? Can you generate these T-cell receptors that define the TCR-T modality? And second, as all cell therapies and frankly gene therapies, the manufacturing hurdle is well to overcome. So what we've done primarily is build a platform around target discovery, TCR asset generation, optimization, which I'll speak to very briefly, and also established, as I mentioned, a GMP facility, which was the local authority late last year. Just to dig a little bit deeper on this promise, I mentioned CAR-Ts, transformative in blood cancers, but they have an extremely limited target space because these Fab fusion receptors, basically antibody fused to some T-cell signaling domains, can only access extracellular targets, as in on the cell surface. Only 10% of the proteome. They have to be very high density because these CAR receptors are not particularly high sensitivity. And people often forget that the CD19 BCMA, they're not actually specific for the malignancy. They're B-cell targets. So there is a target challenge here for CAR-Ts that really hasn't been overcome. TCRs, the native or natural targeting system of T-cells, T-cell receptors, can access 100% of the proteome because they're basically opening up a window and looking inside the cell, because all the proteins in the cell are turned over by the proteasome, they're imported to the endoplasmic reticulum, loaded in HLAs, human leukocyte antigens, and presented at the cell surface. So TCRs are looking inside the cell, and this is how you can open up the massive amount of target space. The question is, why did the industry start with CARs and not TCRs if TCRs are so great? And the question is complexity, and there's a couple of layers here. T-cell receptors are diverse, 10 to the 18 possible diversity in humans. It's like VDJ recombination for an antibody, a similar system without the somatic hypermutation. But importantly, these are cell receptors. They're not antibodies floating in the ether and binding to things. So you have to account for the cell biology complexity as well, which is not trivial. Secondly, the cancer targets themselves. There's a massive universe of tumor-selective gene sequences, mutated sequences, misexpressed. There's just a lot of them to sort through. And there's a second dimension of these human leukocyte antigens. The most diverse genes in humans, different HLAs present different peptides, even if you've got the same tumor-selective sequence. So there's a target challenge, a TCR challenge you have to overcome, all in a cell biology context. The targets are living cells doing their thing. The T-cells are living cells doing their thing with their T-cell receptors. Just to touch on the manufacturing challenge, I mean, no surprise to most people in this room, manufacturing products that are living cells is a challenge. You need specialist infrastructure, you need know-how, there's a high COGS to overcome, and there's lots of competing technologies that are emerging. And just to speak to our solution to address these key challenges, the target hurdle to entry, the TCRs you need to leverage those targets and the manufacturer. So first of all, we've built the platform, which the company's built around. We replicate T-cell biology in the lab by programming human cells. Everything we do is a cell-based assay. There's no guesswork. We let the biology tell us the answer. We built a software ecosystem around this, basically an enterprise software for us to do all the complex database analysis, even logistics day-to-day. We've used this to build this pipeline that I've mentioned, and I'll show that briefly. And we'll be putting that into clinic first time late this year. And like I said, it's important to have that manufacturing infrastructure, process development teams to deploy an autologous gene-edited product, which we're doing with a CRISPR-Cas gene editing system that we announced a couple of weeks ago we licensed from Amendo Bio. So just to touch on, again, the concept behind the platform is, and I mentioned it already, the key is that everything we do is a cell-based assay. No guesswork. We just program target cells and T cells as reporters and let biology tell us the answer, which is the key philosophy behind what we do. And it sounds almost ridiculous that we're using cell biology to make cell therapies. It should be obvious, but it's not the norm in the TCR space today. We do this with a range of biotechnologies, both molecular biology and molecular genetics technology, which basically allows us to compile these analyte cells very precisely, efficiently, reproducibly. I won't talk about any of that today. It's some cool stuff. Please ask me afterwards if you're interested. And the software ecosystem, I said already, we built that in-house basically to account at the base level for T-cell biology reality. It's very complex data. It's very relational data. We're building it to manage all the data pipelines, data workflows, visualization, interpretation. We even build out and manage our laboratory protocols in there and do our inventory and logistics, everything there, basically an enterprise software. Just to touch briefly on the pipeline itself, first assets there going into clinic will be KRAS mutant targeting on multiple HLA restrictions. The CTAs here in Europe will go in around summertime, ideally enrolling end of year, likely probably early next year. It's a big pipeline. People react when they say, oh, you're a biotech, 100 people, you got a pipeline. The explanation is there's a lot of T-cell targets out there, and these aren't 40 independent products. There's a unity of mechanism of action. You're redirecting cytotoxic activity of T-cells as a unity of manufacture. All you're doing is changing the gene construct you're integrating. Basically, we're doing these manufacturing proofs early in the discovery process. We're targeting under 12 months from interesting gene sequence to preclinically validated TCR-T, and there's a momentum to gain here, and economies of scale in clinical programs as well by activating clinical sites, and of course the overhead of our manufacturing. So just to look at some of the very broad detail about the platform, we go about this very differently. We start with a gene sequence of interest. In the oncology space, that's a tumor-selective gene sequence, so a KRAS mutation, a major prime, these CTAs, or a viral open reading frame in virally transformed cancers. Our first step is antigen discovery, or basically HLA-restricted peptide mapping. And people often think that the target is the gene sequence, is the mutant KRAS. It's not. It's the combination of the HLA restriction in which that peptide is loaded and presented at the cell surface. Everyone in the room has a different HLA complement. Your HLAs present peptides differently, even from the same target sequence. So you need to map and identify those peptides very, very precisely. You need to quantify them as well and rank them. Second step is TCR discovery. So we use naive cells from healthy donors. It's just a couple of weeks. It's a cool process where we interact them with these healthy T cells with engineered antigen-presenting cells in a polarizing activation, capture those sequences, the TCRs. We pop these into basically reporter T cells, back validate those to the original target-presenting cells and orthogonally across cancer cells. So this block takes just a couple of months to go from interesting target sequence to not only validated targets, but a candidate list of T cell receptors to work with. The heavy lift in the platform comes from de-risking. So TCRs are promiscuous. They have a propensity to target things they shouldn't. So basically what we're doing here is looking through the off-target space of the human genome, human HLA profiles. Allo and cross-reactivity are the two phenomena that you have to deal with. Basically, we're doing ex vivo thymic selection for people who know T-cell biology. And great, all things considered well, that will spit out some TCR-T assets. Some high-value targets do not have TCRs in the repertoire, in the native human repertoire, and we still want to address those targets. So we also developed a TCR engineering platform, which essentially is a single-step directed evolution of the CDR3 of the alpha and beta TCR chains. I say single-step directed evolution, surprisingly to us, it's just one round of selection in these cell-based arrays, and we can get to more potent TCRs every time. So what you're doing is having a low-quality TCR framework you've pulled from a healthy donor, and as our CTO says, upcycling that to be a clinical candidate. So this is not theoretical. All these modules, these platform modules are novel, they're differentiated certainly collectively, and they're fully operational. This is producing assets all day, every day. I wish I could have time to talk through the data behind this. It's cool data, it's very rich. Ask me afterwards. But the end result, just looking at our mouse study for the G12V, KRAS mutant targeting asset that's in an HLA-A11 background, we saved some mice, we cleared tumors very rapidly in a lung cancer engraftment model. Survival advantage, everyone likes to see it, so there you go. Just finally, on the infrastructure, the manufacturing part, we converted a former AstraZeneca pilot GMP facility near Stockholm to a cell therapy manufacturing plant. This was certified by the Swedish regulators in 2023 and basically enables us to develop very advanced processes, keep change controlling that as better technologies, reagents, equipment come online. I said yesterday on the panel, and I say everyone who listens, nobody has an optimal cell therapy manufacturing process. You have to update as best you can as you go along. We're very committed to do that. We have a number of teams in-house assessing technologies, bringing those to process development, and we'll as we go for our product families. Remember, I said earlier, there's a unity of manufacture, and that's the key here. To scale out across the TCR-T target space, so the genuine T-cell target space, you need to deploy a lot of products. You need to deploy franchises. These aren't monolithic. One product shoots everyone. These are going to be libraries, franchises, warehouses of products, whatever you want to say. I mentioned earlier, we in-licensed gene editing. This is one of the ways in which we're going to be scaling out the manufacturers, standardizing on a gene editing technology, and all we're changing in each manufacturing process for these different asset candidates is the genetic construct that we're integrating into the autologous T cells. And like I said, we've got all the teams in place to do this. It's the main R&D energy in the company right now is manufacturing. The platform's built, it's being operated. All our top scientists are working in manufacturing tech right now. And these conferences are great for pair partnering. We're interested in, of course, co-development perhaps of some of the pipeline assets. There's too many for us to eat right now. We're peeling off top half dozen or so in the first instance. Manufacturing technologies, as I said, is a big focus of ours. DNA technologies, vaccine platforms for reasons I didn't explain today, allogeneic effector platforms and effector cell enhancement, all to us. Also looking to partner co-develop in different disease areas, infectious disease, autoimmunity. Everything I spoke to today is, for those who know T-cell biology, is HLA-1 restricted peptides and CD8 biology. We have a very similar platform for HLA class 2 restricted peptide mapping and CD4 cells. So that's more applicable to infectious disease and autoimmune disease. So finally, with 1 minute to go, big thanks to the team. Like I said, 115 people. We have over 35 nations represented. It's a cool place to work, and a bunch of capacities. We've got mainly biological scientists, but a growing team of software developers, engineers, and all the support staff around that as well. We're based in Södertälje, just south of Stockholm. That's in Sweden, for those who don't know. And yeah, thank you very much for your attention. And,